So this is basically our R&D lab where we do all of our non-GMP work. We do all of our pilot scale and our laboratory bench scale work here. All of the studies for QBD type experiments to support eventual scale up or take place in this particular area. In here, what you're seeing is just a typical pharmacy operation where we have all of our different excipients that we use to weigh out for the particular batches. If we move on to this area here, this is where we do a lot of our liquids. Benchtop liquids processing is in here. We have rheology equipment. We have optical microscopy in here. That's all for semi-solids, creams, and lotions to support those scale-ups. As we move over into here, we do have a trimix that we have. It's a 25-gallon tank. We use this for any of our pilot scale operations before we transfer them out to the other facilities. We have liquids manufacturing in both South Carolina as well as Los Angeles. And so all of the work that's done here supports that tech transfer out to those sites after formulation development is done. We move into this area in here. Again, you'll see a variety of smaller scale equipment, both on the laboratory side and on the pilot scale side. We support all types of granulation, whether it be fluid bed granulation. In this case, you see high shear granulation on a small scale. This is 4-liter, 8-liter type equipment here for batch sizes below 1 kilo. We do go up to pilot scale where we have a 50-liter high shear mixer, which is typically batch sizes around 10 kilos, 12 kilos, something of that nature. We also do fluid bed granulation. This is more of a pilot scale machine that we use. We have both a 7-liter and a 14-liter insert into this. We can do top spray, we can do bottom spray, we can do Worcester coating, we can also do rotogranulation as well.
Which also replicates eventually where it's going to be manufactured in commercial on a smaller scale.
Right. So okay. all of our developmental studies will be done on this smaller scale equipment. In here we have blending and a smaller scale of our dry granulation. We do dry granulation as well. This is a benchtop dry granulation, but we do have pilot scale with a TFC 220, which does about 20 kilos an hour. And then we move up to our IR 520, which is commercial scale, which does about 100 and 120 kilos an hour. Any of our blending operations, obviously blend uniformity, blend homogeneity is the key behind any one of our processes. And we can model it quite well in this piece of equipment here. It's about 12 to 15 kilos. That we feel is really good for a jumping off point. Any smaller than that, we tend to not have a great correlation with our larger scale blenders down the street. So we like to stick at about this size to really get our blend speed and blend times locked in. We move on into compression, which is kind of part of any solid dose operation. We do have a couple of tablet presses. We can do bilayer tablet press on the pilot scale side. We can also do anything from a B and a D tooling, small tablets to larger tablets. But the key behind this operation is it's instrumented with a aftermarket instrumentation package on it that will tell me the pre-compression, the main compression, the injection ports. And we also have a built-in tool that allows us to marry up any of the results we get here based upon the diameter of the turret with any of our manufacturing presses for a seamless transition into the manufacturing area. So once I dial in the compression profile in the laboratory setting, it's almost seamless to transfer it down the street. This is where we do all of our modeling for any of our coatings, whether it's just a simple cosmetic color coating or if it's a functional coating. We can do it in this particular machine. It's very scalable because of the nozzles that it does incorporate. They are slit nozzles, which is what we use down the street. So whether I'm doing a weight gain study for a modified release product, or whether I'm just simply doing a weight gain study for a color coat, this gives me the optimal parameters for running down the street.
So this basic setup is basically designed for developing new products and determining their performance, so to speak, or characteristics and profiles prior to going into commercialization, right?
That's correct.
Okay.
Doing all the background legwork to what you really want to do on the R&D scale is you want to understand your product and your process, because every product is different and every process is different to give the ultimate quality of the product. and everyone has to be assessed individually. And you want to gain all that knowledge, obviously on a smaller scale where it's much quicker and it's much cheaper due to sheer volume sizes than to actually move it down the street and do a large scale study. So it's better, and the FDA under their QBD guidances, they want you to do this. They want you to understand everything up front on the small scale. Take those learnings, and we understand that they're going to change a little bit once you move down the street and get into a commercial environment. But to get the learnings from that to create a science-based approach to how you're going to develop that commercial process, and then they want you to monitor it afterwards through statistical process control. And anytime you need to make a change to that because your data is leading you that way, they allow you to do that sort of thing.
Cool.
Analytical?
Yeah. I'll put it on pause right now. Like I said, we're going to edit it and put it together. The idea is last time we did these virtuals, like I said on the phone, we did it live. And it didn't work badly. It just had a lot of hiccups in it. This way we can prevent any mistakes. And we're all sitting in the same virtual space. And
so obviously at the heart of any development operation is the analytical resources and the analytical lab to be able to test that product. You can make the best product you want, but the problem is you got to test it first to know what you have. And our lab is fully equipped with all the latest state of the art. We have actually undergone a massive upgrade in recent months.
which is an important point to point out to people is that the key to a good CDMO, are they staying current technologies, right?
Yes, we have currently, we're installing new Agilent 1260 HPLCs. In an analytical environment, it's very critical that we also have them with diode array detectors that allows us to see molecules in a 3D type of arrangement, so we can see where all these ellipses pop up. So we have that. We have brand new GCs that we've just invested in, all to stay current. Particle size analysis, we do that for everything. We're currently doing an experiment right now. We're checking the particle size of some things so that we can determine whether or not to make a switch on a manufacturing process or not. In here we have, this is our DISO room, where all of our drug releasing takes place. So anytime you create a drug product, they want to know what the drug release is going to look like. We mimic that in vitro through the use of dissolution apparatus. And all of this is current equipment. We can do apparatus 1, 2, and 3 down at the end of the bench.
Okay.